Our group is method separation and purification. So we develop methods at the analytical as well as at the preparative scale. And this, I'll be talking about this at several points in my presentation. Some of the complexities we encounter when doing modeling, chromatography modeling, is working with closely related components, which are hard to separate. Also, looking at developing purifications methods, purification scale methods based on analytical scale. Another challenge is working with complex mixtures that are beyond the ability of one-dimensional liquid chromatography to separate. And the last example here is understanding how to model large biomolecules correctly when using cryotropic reagents. So the first two parts I have presented previously, so I will be talking about very briefly, while I focus most of the time talking about the multi-dimensional separations and the protein work. Now, again, this is the traditional workflow. And when we have closely related components, we try to build a workflow around that, as I will specify in the next slide, where I have a set of 20 nucleotides. In a typical example, we usually try to separate 5, 6, maybe 7 of these nucleotides. And every time we're asked to separate, to determine most of the time the percentage of each of these nucleotides, we develop a fit-for-purpose method. Rather than going through this exercise every time we receive such samples, what we did here is we built an optimized generic method in which we could separate all of the components and all of the nucleotides here. So this single method over here can separate the 20 nucleotides, taking into consideration that most of the time we don't really need to separate all of them. We have 5, 6, and this was related to a certain program. And we have excellent match between predicted and experimental time, retention time, as you could see in the difference in the retention time here. Now, this is great because it simplifies the work and the time needed to generate these methods. This is half of the story, because we're also using this workflow, this database, to develop preparative scale methods for purification. And this is related to the second point. How do we look at developing purification methods based on analytical methods? Now, to answer this question, we need to understand what's the difference between a good analytical method and a good preparative scale method. And this is, again, the nucleotide example. In an analytical method, we look at resolution between all the peaks. While in a prep scale or a method suitable to scale up, we are targeting one analyte, where we load that analyte, separate, purify, and what is very important is we have resolution around the peak of interest, and we don't really care much about the other peaks. So we tend to shorten the runtime, as you see here, 17 versus 60 minutes, and there's re-equilibrium. And there's more resolution around the peak of interest, which leads to higher productivity, higher loading of the material. And there's some simple math here of the enhanced productivity, and this results in an increase of 60-fold, meaning that if it takes you a week to do the purification using this method, if you scale it up, it actually takes you like an hour to do it using the optimized method. This is a great example for prep scale. Now, this is a real example in which we did actually use it for the nucleotides. Peak of interest was peak number 2, as you see on the left side. This is the analytical scale. This is the database we showed in the previous slide. We re-optimized the method to have more resolution around peak number 2, and this is what we get. A lot of more resolution around peak number 2. We can scale it up, and we could easily load more of that component and isolate it and establish the structure or confirm the identity. And the increase in productivity was 200-fold. Again, what takes several months could take one day here.